Hello everyone, here we are going to discuss about the classification of enzymes and nomenclature of enzymes. Okay, classification is nothing but categorizing the different types of enzyme and then you have to put them in a specific class. Okay, the scientists have isolated and discovered more than thousands of enzymes from different organisms and then they classified them. Okay, into or like under 6 major classes. Okay, we'll talk about that. Is actually the main concept is all about. Okay, then nomenclature of enzymes means giving a name to the enzymes based on the function it does. Okay, in a living organism, what kind of function it does in the living organism, based on that the naming of the enzyme is done. That is the procedure of nomenclature is done. Okay, so let me just write. The scientists have discovered more than thousands of enzymes. Okay, how many? More than thousands of enzymes. It's a huge number actually, from the cells. Okay, this is the cell now, tiny cell. Okay, from the cells of different organisms. That organisms are the bacteria, even the fungi, even you can also write algae, even you can also write plant. Okay, so here we write animals. Okay, from these different types of animal cells, they have extracted means removed the enzymes, thousands of enzymes. Okay, so that is actually the thing they did. And after that, based on what type of chemical reaction they does or what type of chemical reaction they catalyze. Okay, so what type of chemical reaction they catalyze here we'll write here the enzymes, they in the sense enzymes in the living organism. These enzymes are classified under 6 major types. How many? 6 major types means classes here. Okay, so we write the 6 major classes. I'm going to help you here with a small code so that you can remember it. You are going to consider the first alphabet of all 6 major classes, that is O, the first class, second class, third class, fourth class, fifth class and sixth class. So we have 6 classes here, our first alphabet of all 6 classes. So how do you read this? Utlil, remember this, done. Utlil is the code to remember. Okay, so I'm just helping you here. Okay, very simple, because this is very important for your competitive, so you need to remember this in order. Utlil is the order. Okay, fine. So these are the 6 major classes. Now let me just write each class. I forgot. Okay, so I said 6 major classes. If you look into one class, for example, if I pull out this one class, that is O here, if I just pull out oxidoreductases, and in this one particular major class, you are going to see about 4 to 13 subclasses. 4 to 13 subclasses are there in each major class. So here I am not going to discuss about the subclass. Okay, it was the session going to be very lengthy and it is not necessary for your competitive part also. Okay, you are going to study in the higher classes. Okay, so we just concentrate on this Utlil. Okay, so we go with the first major class of enzymes, that is classification itself. Okay, so what is it? O. What is O here? O is oxidoreductases. Oxidoreductases. In the name itself, you can easily identify what type of chemical reaction they bring in the metabolic process, that is oxidation reduction. Okay, oxidoreduction. It is also, this particular major class, oxidoreductases, also called as dehydrogenases. Okay, so we write here dehydrogenases. You are a bit familiar with this word, right? Dehydrogenases. It's very common actually in all biology chapters. Dehydrogenases enzyme is very common, but oxidoreductases is not that common actually. Okay, because this is easier to pronounce and easier to understand, but whatever it is, both are same actually. They belong to the same class, the first class. Let me just talk about its function, oxidoreductases or dehydrogenases function. For example, just let me write a substrate. A chemical reaction is going on. Okay, so let us just put a plus here and we just put one more here. What is this S? S is a chemical, that is substrate for the enzyme. Substrate. S is one substrate, and here another substrate is also involved in the chemical reaction. So there are 2 substrates, and this S is in its reduced form. Okay, so we just write this S is in reduced form, and this S is in oxidized form. Okay, so there are 2 substrates, both are in different form. One is in a reduced form, other one is oxidized form. Okay, so now what this particular enzyme does, so if I just if I just add this particular enzyme oxidoreductases to this chemical reaction, and they are going to bring a very different product, that is, yes, earlier it was reduced form, now they become oxidized. One substrate now become oxidized from reduced form, whereas the other one, so what is the other one, that is our green color substrate, S dash, that is actually earlier it was oxidized, but now they become reduced. Okay, so this is actually the main function of oxidoreductases. Got it? So they are going to bring reduction as well as oxidation reduction. They, hence the name is oxidoreductases or dehydrogenases. Got it? Between the two substrates. This is actually the first major class, oxidoreductases. I hope that you understood this. It's very simple actually. We go to the next one, that is second one, that is T. What is T here? Transferases. See, very simple. Transferases, in the major class name itself, the function is given. They are going to bring transfer of what? Transfer of a group between the substrate. Okay, so we'll see here. The main function of this enzyme is they are going to bring the transfer. They are going to change transfer of a specific group. Okay, so between the substrates. If two substrates are there, they are going to shift the group from one substrate to the other substrate. Okay, we see that thing here, and let me just write yes is one substrate here, and let us just put a group. So this particular substrate, a particular chemical, a group is attached. Okay, this is the substrate now, and now what are you going to see now? And this is what is the second substrate. S dash is the second substrate that is actually involved in the chemical reaction, which is catalyzed by transferases enzyme. So let us assume that now the transferases enzyme, I'm going to add this transferases enzyme to this particular chemical reaction, and you're going to see shifting of this particular group to this particular substrate. Okay, so yes is going to lose the group. Yes. Okay, so and now S dash, earlier there was no group actually attached to the S dash, but now it's going to gain it, so that is nothing but shifting. Okay, so S dash now gets that group which was attached to S earlier, okay, before this particular enzyme came into act. Okay, so this is actually the two different types of major classes and its function. I just want to add one more very important thing. Let me just tell you, see, shifting of group from one substrate to other substrate is possible only other particular bonds are there except hydrogen bond. Okay, except, so this particular function is possible when hydrogen bond is absent in any of the substrate except hydrogen bonds. Okay, so what is required for this particular mechanism or function of transferases? Hydrogen bond should be absent in the substrate. Hydrogen bond should be absent. Got it? So this is very important. Please note this point. Very important actually. Okay, so hydrogen bond should not be there in the substrate. Okay, let me just erase it and go to the third enzyme major class. Okay, the third class is actually ours, H here from Othlil, that is hydrolases. Hydrolases. What is this hydrolases? Again, in the major class name itself, we can easily identify the function of hydrolases. That is, they are going to bring hydrolysis, that is breakage of the bonds. Hydrolysis. What is hydrolysis? With the involvement of the water. So water is required here. Okay, an enzyme actually breaks the bond, okay, in a particular substrate or between the substrate. Hydrolysis. They are going to bring the hydrolysis. Let us see what type of bonds they can break with the help of hydrolysis. Okay, the hydrolysis enzyme is now going to break different types of bonds that can be written as the very familiar bond, that is peptide. Where do you see peptide bond? You see them in polypeptide chain. Okay, so we go to the next one, that is even in the glycosidic bond also you see. Where do you see glycosidic bond? In the nucleoside of the DNA, and even the ester bonds and ether bonds. Okay, so even you see between CC bonds also, and even you see between the halides also. and even you see them between p n bonds also. Okay, so whatever the bonds I have listed here, these enzymes going to bring the hydrolysis. What is hydrolysis? They are going to break the bonds. Got it? When the bonds are broken, obviously the products will be produced in a chemical reaction. Okay, so this is the main function of hydrolysis. Let us just go to the next class, that is, I use red color, that is actually called as liases. Liases is actually our fourth major class. The fourth major class. What is the main function of this? Very simple. Let me just use blue color because I have already used green color here. So removal of groups. Okay, so liases actually does, they just remove the removal of a particular chemical group which is already attached to a substrate, that is from substrate. So in the chemical reaction, substrate is there, and to that substrate a group is attached. Okay, so they are going to break the bond that is actually between the group and the substrate. They just break the bond, that is detaching the group from the substrate. That's it. But remember here, hydrolysis is not possible. So let me write that also. Hydrolysis is not possible because these liases enzymes are not hydrolysis, correct? So they are going to break the bond between the group and the substrate with the help of other mechanisms. So let me write here other mechanisms. Okay, some other mechanisms. So this is actually the main thing. Let me just write the example for it. So that is, for example, CC bond is there, and to that X and Y are the two different groups are attached. Okay, and now I'm going to add this enzyme, that is liases to this particular reaction. Actually, let me just write L here, really, liases. Now they are going to bring this, see, so the groups are being removed now. It is no more attached to CC bonds, correct? Okay, and you see the remaining CC bonds here, but the CC bonds, okay, I should have used red color actually. Okay, red color, CC bonds here, got it. But earlier it was having only single bond, but in this case double bonds are there, correct? So that means I'm going to add one more statement here. Removal of the group, a particular group from the substrate other than hydrolysis using other mechanisms, leaving double bonds. So we'll just add here. So whenever liases are in work in a particular chemical reaction, or when they are catalyzing a chemical reaction, they not only separate the groups from the substrate, but also leave double bonds, DB, double bonds. That's what you see here. Got it? Okay, so this is actually the fourth major class. Let me erase it and go to the next one, that is okay, so the fifth one, let me just write the fifth major class of the enzyme, that is LI. Yes, isomerases. In glycolysis these enzymes are there, correct? Isomerases enzymes are very common actually. Okay, so let me just write what is exactly the function of isomerases. They are just going to bring the interconversion. Okay, what is interconversion? Just change of position, that is interconversion. That is nothing but change of position, interconversion of optical. I think you are a bit familiar with optical isomers, correct? Okay, optical isomers, even geometric isomers, and even positional isomers also. Okay, so they are going to bring the interconversion within the optical isomers, geometric isomers, and positional isomers. Got it? Okay, so let me just give you the example for optical isomers here. So we just write the example here, and that is here COH, and here we have OH, and here we have CH3, and here we have H. Okay, so this is actually optical isomer. And now when you add this isomerases enzyme to this particular chemical reaction, and you are going to see the interconversion of some groups. Okay, so you could see here, let me just put a circle around so it will be easy for you. Okay, see here, this becomes H3C, and this particular now shift, correct? See what has happened to this particular chemical now. It is now optical isomers. You could see the mirror image of it. Got it? So shifting, This is actually the main function of isomers in optical isomerases enzyme in optical isomers. Okay, so this is one thing. Let me erase it and go to the geometric isomers. One more example if it is required for you. So that is, we have C double bond C, and here we have CH3, and here we have H, and here we have same CH3, and we have H here. Okay, so here you are going to see the change of the groups. So that is it, this side remains the same, but this side you could see H, and this side Ca3. What you have seen here is change, that is actually interconversion, correct? So there is change of the position of these particular groups actually. So this is geometric isomers. Got it? Okay, so isomerases bring the interconversion of groups within optical isomers, geometric isomers, and positional isomers. Okay, so we go to the last one, that is actually our sixth and the last major class of the enzyme. That is, let me just write the sixth type, that is we have here is L, that is ligases. These are also famously called as molecular glues. Okay, you will come across with this ligases in genetics. So that is molecular basis of inheritance chapter. Okay, molecular glues. They actually bring the two different chemicals and stitch them. Okay, they are nothing but, link the two different groups. Okay, so the function, let me just write here. They are going to bring the linkage, that is they link the two different groups. Okay, so let me just tell you the groups actually, so or compounds. Okay, so even I can write this as compounds also. Okay, fine. So the compounds could be like having C-O bonds, or even CS bonds, or even CN bonds, or even PO bonds. So this is actually the main function of ligases enzyme. Got it? Okay, so they are going to link the two different types of compounds or groups, okay, in a chemical reaction. So they are also called as molecular glues, and they are really helpful in genetics, particularly in recombinant DNA technology. In biotechnology, we're going to study about it. So this is actually all about classification of enzymes. Nomenclature is very simple. OTHL is the thing. So there are 6 major classes. That is actually nothing but O means oxidoreductases, transferases, hydrolases, liases, isomerases, and ligases. So please learn everything and be updated. Thank you very much.